Assalamu alaikum. I am Haiga Ufran of Group A. Me and my group have prepared a presentation on experiment number 5, fructose and mannose. And the section B, procedure for the detection of fructose and mannose, have been prepared by me. So firstly, I will give you a presentation on the procedure for the detection of fructose. For the detection of fructose, we have to perform different tests, and to perform these tests, we require test tube holder, ethanoic alpha naphthol, concentrated H2SO4, iodine solution, Benedict reagent, Barfoed reagent, Salmonov reagent, and burner. First, we will perform Molish test. In Molish test, we have to add 2 milliliter of carbohydrate sample in a test tube and add few drops of ethanoic alpha naphthol in it. Then we will pour concentrated H2SO4 along the side of test tube. After that, we will observe that purple ring appears, which indicates that carbohydrate is present. Then for iodine test, we have to take 2 to 3 milliliter of sample in our test tube and add few drops of iodine solution. When we will perform iodine test, we will observe that transparent yellow color appears, which shows that polysaccharide is absent, means either the monosaccharide or disaccharide is present. After performing iodine test, we will perform Benedict test. In Benedict test, we will take equal volume of both the sample and Benedict reagent in a test tube, and then we will heat it using burner for about 3 minutes. We will observe that red color appears within 3 minutes, which means that it is a reducing sugar. After that, we will perform Barfoed test. In Barfoed test, we will take the equal volume of each the sample and Barfoed reagent in a test tube, and then we'll heat it for about 5 minutes. We will observe that red precipitates are appeared before 5 minutes, means the sample is a monosaccharide. Then to perform Salmonov test, we will take 1 milliliter of sample and 2 milliliter of Salmonov reagent in a test tube and boil it. By doing so, we will observe that cherry red color appears, which shows that ketose is present. All these tests will indicate that the sample is of fructose. After that, we will form the osazones of fructose. To form the osazones of fructose, we will take 4 to 5 milliliter of sample in a test tube and then add few drops of phenyl hydrazine in it and mix it. Then we will keep the test tube in a boiling water bath for about 20 to 30 minutes. And after the formation of yellow or orange crystals in a test tube, we will pour it over a glass slide and observe it under the microscope. When we will observe that, we will see needle or grass-like crystals are formed. So that was the procedure for the detection of fructose.